The overall goal of the following experiments is to measure the mechanical properties of the interphase cell nucleus and its physical connection with the surrounding cytoskeleton. This is achieved by applying mechanical strain to cells plated on a transparent silicone membrane and imaging the induced nuclear deformations. Increased nuclear deformations correspond to decreased nuclear stiffness. Since changes in nucleo-cytoskeletal coupling can also influence the induced nuclear deformations, intracellular force transmission between the cytoskeleton and nucleus is directly probed by applying localized strain to the cytoskeleton. The displacement of fluorescently labeled nuclear and cytoskeletal structures is then imaged. Results are obtained that display the stiffness of cell nuclei relative to the surrounding cytoskeleton based on the substrate strain experiments. The microneedle manipulation assay reveals that disruption of nucleo-cytoskeletal coupling causes defects in intracellular force transmission.
The main advantage of these techniques over existing methods, like micropipette aspiration of isolated nuclei, is that they enable the measurements of nuclear mechanics in intact living cells without disturbing normal cellular architecture.
These methods can provide insight into how changes in nuclear envelope composition can influence mechanical properties of the nucleus. They can also be applied to study the role of nuclear mechanics in the context of diseases, such as laminopathies or cancer.
Each strain dish consists of a custom-made bottomless plastic dish with a diameter of 3 inches and a plastic O-ring to hold a silicone membrane, which serves as the cell.